Hi everyone, my name's Hayley.
Hi, I'm Maya, and today we're going to talk to you about CRISPR.
Imagine a famous author who just recently released their latest and greatest novel that has long been awaited by the public. Many people are super excited to read it, the editors love it, the publishing companies pass it and mass produce it to make it available in bookstores worldwide. But once the readers start diving in, they notice that a few of the pages are missing and there's a whole hole in the middle of the plot.
How would you fix this situation then if they're already all spread throughout?
I feel like it would be pretty cool if we could travel all around the world super fast and just add the missing page to every book so that the readers are not lost. What's even cooler is that DNA can be compared to a book in that way. It's your genetic makeup that makes you who you are. Everyone has it because it's our body's means of directions to tell our cells each and everything that they are supposed to do. All of your DNA came from that of a single cell, and each is unique to you. Once that genetic material is copied over and over again, one cell splits and becomes trillions of cells, all with the same exact DNA. This ties back in with if how there were to be a mistake in the DNA, that mistake would be copied over and over again into each and every copy of the cell. These mistakes are called mutations in the body and can lead to diseases like sickle cell anemia, cystic fibrosis, and even cancers. Until recently, these mistakes could not be fixed, and the symptoms of these diseases could only be treated. What if we could send a healthy copy of DNA into each and every one of the cells and remove the part of the DNA with the mutation? This is actually possible with CRISPR.
So what is CRISPR? CRISPR stands for Clustered Regulatory Interspace Short Palindromic Repeats. There are two major components of guide RNA that go into CRISPR. There's gRNA and sgRNA. Along with these guide RNAs, there's an associated endonuclease known as a Cas protein called Cas9, which causes gene knockouts. The overall steps to CRISPR are that the guide RNA recognizes the target DNA region of interest and tells the Cas9 where to go to make the edits using gene knockout. So to go over a basic step-by-step of CRISPR, there's a Cas9 finds and forms a complex with the guide RNA. Then the complex made attaches to its complementary base pair that is near the spacer. Following that, the Cas9 complex, which is an endonuclease, so it cuts the double strands of the DNA, and the new programmed DNA is inserted into the cut from the endonuclease, which you can see happening in both of the images listed. So, as we've talked about CRISPR, there's a timeline that goes over the step-by-step, which we will go through following this, but just want to give you a background before we go into more detail. So CRISPR started its first discovery in 1987, which was found in E. coli. Following this, there's been many evolutions and breakthroughs that have happened over time, but into today's world, there's Cas9 that is used for epigenome editing. So from here, Hayley's going to talk to you guys about the major history behind CRISPR.
So the studying for CRISPR began in 1987 when a Japanese scientist, Yoshizumu Ishino, and his team began to ask questions. They accidentally cloned an atypical region of repeated sequences interspersed with spacer sequences when they were looking for a gene responsible for the conversion of alkaline phosphatase. They did not have a lot of information on the DNA that they found, so this finding went under the radar until 1993 when J.D. Van Ebden discovered that Mycobacterium tuberculosis, or more commonly just referred to as tuberculosis, has different strains that have different spacer sequences between the repeats. Over time, these spacer sequences were found more commonly and were named CRISPR by Francisco Mojica and Ruud Janssen in the early 2000s. These researchers noticed CRISPR-Cas complexes serve as a defense mechanism from viruses to prevent repeated infections in the future. In 2012, Nobel Prize winners Jennifer Duanda and Emmanuel Carpentier discovered that CRISPR-Cas9 can use the cut and paste tool when they want to modify genomes. This finding enhanced gene editing as we know it. This application of CRISPR has already made changes to gene editing with helping diseases that are infectious or cancerous. In years before these massive evolutions made with CRISPR, the CRISPR-Cas9 system was found in prokaryotic immune systems that target bacterial viruses to be cut out in order to keep the organism alive. This practice, as previously stated, is being worked into allowing genes to be cut and pasted into other organisms, which is used to possibly aid genetic disturbances.
So let's talk about these specific evolutions that CRISPR has given to the world as we know it to be. So to begin, gene editing, which in the form of therapies has grown as the first trial of the original CRISPR cell therapy with sickle cell disease in 2019, along with generating a receptor in the immune system cells of cancer patients. Also, in recent years, COVID has run wild. We've constantly been in a world where we're hearing, test if you're feeling ill, test if you're feeling ill, test if you're feeling ill. So where are all these tests coming from and what are these tests? So in 2020, if you remember, tests were very limited. This is because CRISPR had not yet formulated the technology that it has in recent years, such as 2020 and 2022, where these tests had become more abundant because CRISPR is there to help aid in the process. So what are these tests? What does CRISPR do to these tests? These tests use Cas9 to detect if the pathogenic COVID-19 gene has invaded the eukaryotic system, which is any system that has a nucleus inside of their cells. In agriculture, scientists are working to have CRISPR-modified foods that are resistant to all things harmful. We will talk about these later in tomatoes found in Japan. Nonetheless, with recent talk and the worries of our climate, CRISPR can be used for bioenergy that can help keep our lives as we know them to be today when our resources begin to fade away in the coming years.
With every new technology, it takes years or even decades for the general public to adopt and accept it. The same thing is happening in science with CRISPR as new information is found. People, meaning researchers and the public, don't like change. So when it is proposed, extensive evidence must be found to indicate its usefulness and a successful means of production. For CRISPR in particular, there are many fears among both the scientific and general public that can be combated with a further dive into the facts. Let's talk about a few of these common misconceptions. The first misconception that we're going to address is that CRISPR and genetic modifications are the same exact thing. Truthfully, genetic modification actually introduces foreign DNA from one organism into another, therefore modifying the DNA. Furthermore, this foreign DNA can come from effectively any species of organism, bacteria, plants, insects, you name it. On the other hand, CRISPR is simply gene editing where existing DNA is cut, fixed, and pasted back together. This misconception came from people thinking that all changes to genes are the same and generally are not good things. People are fearful of science that they don't understand, but learning the differences and how many of these technologies are used for good can ease that fear.
Another misconception that CRISPR has brought is that CRISPR isn't precise and will cut anything. Well, we can see where people are coming from with this. It's not accurate. The truth behind this is that Cas9 can find its target gene down to the single bases, which are adenine, thymine, guanine, and cytosine. From there, it will rewrite each individual base to create a sequential order that makes up an entire gene. Once the gene is rewritten, CRISPR will stop. It will glue the strand of DNA back together, and this edit will make the sequence flow as it should. This misconception stemmed from how genetic modification is a much broader approach and is often a shot in the dark. CRISPR can function with specificity, and new technologies have allowed it to be accurate down to the single base.
The next misconception is that CRISPR is highly unregulated. The truth behind this misconception is that CRISPR is currently under the same regulations that genetic modification has, but the regulations for CRISPR will likely increase as the popularity of the method increases. The seemingly limitless possibilities of this technology can be intimidating. However, strict regulations are in place to ensure that researchers are using it ethically and under supervision. For example, there is speculation that designer babies or children with DNA edited to have desirable traits will be created. In this situation, parents would be able to choose exactly how they want their children to look, how tall they are, what genes they have, and that can lead to many complications and faces a lot of ethical issues. While research on this possibility may be conducted, it doesn't quite align with the intention of this technology. Reversing diseases is the goal of the research and what majority of its efforts will be focused on moving forward.
So following the other misconceptions, one of the main misconceptions that are among many new scientific findings are that it is more expensive. To this, CRISPR is expensive and money should be allocated elsewhere is commonly heard everywhere. However, this is false. The truth behind this is that CRISPR is more affordable than other bioengineering techniques, and there is clearly a need for some means of modification, as the results promised can have significant benefits. CRISPR, as we have already discussed, has the ability to prevent disease, increase food production, help aid in bioenergy, and it can help with already found diseases in the future. With that said, while most new technologies are expensive due to the abundance of materials needed, the only materials needed to complete a CRISPR modification are the correct RNA fragments to code for the desired gene and the enzymes that will produce it to the correct location, or the Cas9 protein. Those are two necessities compared to thousands upon thousands with many other bioengineering techniques. This misconception is rooted in the ongoing issue where many people associate new technologies with increased costs. But with CRISPR, there is not a need for fancy or expensive lab equipment. All you need is DNA. So now that you've heard all, not to believe what you hear and see online about some of the cool trademarks that CRISPR has accomplished. To begin, on September 16th, 2021, the FDA approved research and trials to be done using CRISPR that work to find a potential cure for HIV. Scientists do this by using a one-time gene therapy in patients who are HIV positive instead of the lifelong treatments that individuals that are HIV positive would normally endure. To do this, the Cas9 nuclease cuts out the HIV that is around the DNA, which essentially removes the virus individual that is HIV positive, HIV negative.
The second newest advancement in CRISPR technologies happened on September 24th of 2021, where the first CRISPR edited food went into the market in Japan. It was a tomato, and these tomatoes were edited to contain high amounts of GABA. This is desirable to consumers because of the health benefits associated with this additive. Oral intake of GABA can help combat high blood pressure and increase levels of relaxation. There are many other GABA-enriched foods on the market, so the people in Japan are used to seeing this. So when they saw that GABA was in tomatoes too, they thought, great, this is just another way we can get our sources of GABA. But in those other foods that contain the GABA is added just after, as like a mineral would be added, or vitamins. And we see like Gatorade here in America, it's added in the same way. So that GABA is just added after and is not encoded in the product's DNA. But in this situation with the tomato, this is the only food currently available made with CRISPR. but researchers are hoping to broaden their capabilities and include more CRISPR-edited foods in daily life. After hearing all about CRISPR, there are some important points to remember. CRISPR is useful and exciting.
CRISPR is allowing scientists to target fatal genes and cut them out of the genome using Cas9, which will help save so many lives.
Also, CRISPR is not a harmful mechanism. It is primarily used to trigger immune responses that prevent the good bacteria from being infected by the bad.
These reasons, along with so many others, will enhance the lives of many and change the world to be a better place because of the CRISPR-Cas9 systems in science. Thank you for listening.